Welcome everyone. I'm excited to kick off today's presentation on advanced techniques in biochemistry. On this first slide, you'll see the title Chromatographic Techniques, which is a key area we'll explore today. We'll cover the fundamental principles, different types of chromatography, and some of the latest advancements. So, let's dive in and discover how these techniques play a crucial role in biochemistry research and analysis. Chromatography is a physical method of separation, which means it relies on physical properties rather than chemical reactions. In this process, the components we want to separate are distributed between two phases. One of these phases is stationary, meaning it stays fixed in place, and we call this the stationary phase. The other phase is mobile, meaning it moves through or along the stationary phase, and this is called the mobile phase. The key idea here is that the mobile phase travels through the stationary phase in a specific direction, carrying along the different components to be separated. This process occurs because different components have different affinities or preferences for the stationary and mobile phases. These differences are quantified by what we call distribution constants, which essentially tell us how much of each component prefers one phase over the other. Because these preferences vary among the components, they move at different rates, allowing us to separate them effectively. Understanding this fundamental concept helps us grasp how chromatography works as a powerful separation technique in chemistry and biochemistry. It's all about exploiting these differences in distribution to isolate individual components from complex mixtures. So, chromatography is a technique used to separate and identify the different components within a mixture. Essentially, it works by allowing molecules to distribute themselves between two phases, a stationary phase and a mobile phase. Imagine the mixture as molecules competing to stay in one phase or move with the other. Molecules that tend to spend more time in the mobile phase get carried along faster through the system. This difference in how long they stay in each phase allows us to separate the components effectively. This technique is fundamental in analytical chemistry because it helps us analyze complex mixtures and identify individual substances. Throughout this presentation, we'll see how chromatography plays a vital role in various scientific applications. Let's talk about distribution coefficients, which are really the foundation of all chromatography techniques. The key idea here is the partition coefficient, often called CADE. This coefficient tells us how a compound, which we call the analyte, distributes itself between two immiscible phases. Think of it as a kind of balance or ratio, how much of the analyte prefers one phase over the other. When we look at two phases, let's label them A and B. The distribution coefficient remains constant at a specific temperature. This consistency is crucial because it allows us to predict how the analyte will behave during separation. The mathematical expression for this coefficient involves the concentrations of the analyte in each phase. But what's most important to remember is that CADE encapsulates the affinity of the analyte for each phase. Understanding these coefficients helps us grasp how compounds move and separate in chromatography, which is all about exploiting these differences to achieve effective separation. So, in essence, the distribution coefficient is central to understanding how chromatography works. It's the mathematical and conceptual backbone of how analytes partition and ultimately get separated. Now, let's look at how chromatography is classified based on the mobile phase. First, we have liquid chromatography, where the mobile phase is a liquid. This includes techniques like LLC and LSC, which are quite common in analytical chemistry. The key point here is that the mobile phase moves through the stationary phase in a liquid form, carrying the sample along. Next, we have gas chromatography, where the mobile phase is a gas. This includes GSC and GLC, which are especially useful for volatile compounds. The main idea to remember is that in gas chromatography, the sample is vaporized and carried by an inert gas through the stationary phase. These classifications are fundamental because they help us choose the right technique depending on the nature of our samples and what we're trying to analyze. So, understanding whether your mobile phase is a liquid or a gas is crucial in selecting the appropriate chromatography method for your analysis. Now, let's let's look at how the stationary phase is packed in different types of chromatography. First, we have thin layer chromatography, or TLC. In this method, the stationary phase is a very thin layer that's supported on plates made of glass, plastic, or aluminum. Think of it as a super thin, even coating that allows for quick and efficient separation. Next is paper chromatography, where the stationary phase is actually a thin film of liquid that's supported on an inert support, like paper. This method is simple and effective, especially for separating small molecules or mixtures. Lastly, we have column chromatography, which involves packing the stationary phase inside a glass column. This setup allows for larger sample volumes and is often used for preparative purposes, giving us more material after separation. Understanding these packing methods is key because they influence how the separation occurs and what kind of samples we can work with. Each method has its own advantages depending on the sample size, complexity, and the level of resolution needed. So, as we go along, keep in mind how these packing techniques fit into the broader picture of chromatography and its applications. Let's explore the classification of chromatography based on the force of separation. First, we have adsorption chromatography, where separation occurs due to the different affinities of substances for the stationary phase. Next is partition chromatography, which relies on the distribution of compounds between two immiscible phases, typically a liquid stationary phase and a mobile phase. Then there's ion exchange chromatography, where separation is based on the charge properties of molecules, often used for charged ions or molecules. Following that is gel filtration chromatography, also known as size exclusion chromatography, which separates molecules based on their size, with larger molecules eluting first. And finally, affinity chromatography, which leverages specific biological interactions, such as antibody-antigen or enzyme-substrate binding, to achieve highly selective separation. These different methods are key to understanding how we can target specific molecules depending on their properties, making chromatography a versatile and powerful technique in our analytical toolkit. As we move forward, keep in mind how each force of separation plays a crucial role in tailoring the technique to the specific needs of each experiment or analysis. Now, let's delve into the qualitative analysis. The main goal here is to confirm whether a specific analyte is present in a test sample. We do this by comparing the retention time of the peaks in the chromatograph with those from an authentic reference sample of the analyte. It's important that both are obtained under identical chromatographic conditions to ensure accuracy. Another key point is the method of confirmation via spiking. We take a second sample of our test sample and add a known amount of the authentic compound. If the analyte is truly present, we should see a single peak that increases in area as predicted. This helps us confidently identify the analyte in the sample. Additionally, structural confirmation can be achieved using advanced detectors like a mass spectrometer or an NMR spectrometer. These tools provide structural evidence, giving us a more definitive identification of the analyte responsible for the peak. Connecting this to our overall theme, qualitative analysis is crucial because it not only detects the presence of a compound, but also confirms its identity, ensuring the reliability of our analytical results. Let's talk about quantitative analysis. The main goal here is to confirm whether a specific analyte is present in our test sample, and also to determine how much of it is there. To do this, we rely on the peak area in our chromatogram, which we compare to a calibration graph that we've previously established. Now, the key concept to remember is that the area of each peak in a chromatogram is proportional to the amount of analyte that produced it. So, larger peaks generally mean more of the substance. To measure this, we look at the height of the peak, which we call HP, and the width at half the height, or WH. By multiplying these two measurements, we estimate the area of the peak. This method works well, but it can be quite time-consuming, especially when you're working with complex samples or doing many analyses. That's when dedicated tools like integrators or microcomputers become really valuable. They can perform these calculations quickly and accurately. Overall, quantitative quantitative analysis is essential for precisely identifying and measuring analytes, and understanding the relationship between peak area and analyte concentration helps us interpret our chromatographic data effectively. Now let's talk about column chromatography, which is a powerful technique that relies on differences in protein properties like charge, size, binding affinity, and other characteristics. Essentially, we use a porous solid material, known as the stationary phase, which is carefully chosen for its chemical properties. This material is held within a column. Then we have the mobile phase, a buffered solution that flows through the column. When we introduce the protein sample, dissolved in this same buffered solution, it's layered on top of the column. From there, the protein starts to percolate through the solid matrix, forming an expanding band within the larger volume of the mobile phase. What's interesting is that different proteins will move through the column at different rates. Some will migrate faster, others more slowly, depending on their unique properties. This separation allows us to isolate specific proteins based on how they interact with the stationary phase and how they move with the mobile phase. Overall, column chromatography is a versatile and essential technique in protein purification, helping us to distinguish and isolate proteins efficiently by exploiting their individual properties. Now, let's take a closer look at a typical column chromatographic system, which uses either a gas or liquid mobile phase. At its core, this system consists of several key components that work together to separate and analyze mixtures. First, we have the stationary phase, which is the material packed inside the column. This is crucial because it interacts with the sample components, helping to separate them based on their different affinities. Next, there's the column itself, which serves as the vessel holding the stationary phase. The design and length of the column can significantly influence the separation efficiency. Moving on, we have the mobile phase and its delivery system. The mobile phase flows through the column, carrying the sample with it, and its composition and flow rate are vital parameters for successful separation. Then there's the injector system, which introduces the sample into the system in a controlled manner. Proper injection technique ensures reproducibility and accuracy. Following that, we have the detector and chart recorder, which identify and record the separated components as they exit the column, giving us essential data for analysis. Finally, there's the fraction collector, which collects the separated fractions for further analysis or use. Altogether, these components form the basic framework of a column chromatographic system, and understanding how they interact is key to optimizing separation and achieving reliable results in our analyses. Let's take a look at the two main forms of column chromatography. The first is liquid chromatography, or LC, which is most notably represented by high-performance liquid chromatography, or HPLC. This technique uses a liquid mobile phase to separate components based on their interactions with the stationary phase. It's widely used because of its high resolution and efficiency. The second form is gas chromatography, or GC. In GC, we use a gas as the mobile phase, and it's particularly effective for volatile compounds. Both methods rely on passing a mixture through a column packed with a stationary phase, but they differ mainly in the state of the mobile phase and the types of compounds they analyze. Understanding these two forms is crucial because they form the backbone of many analytical processes in chemistry and biochemistry. They allow us to separate, identify, and quantify complex mixtures, which is essential for quality control, research, and development. So, in essence, liquid chromatography and gas chromatography are the two pillars supporting the broader field of separation science, each tailored to different types of samples and analytical needs. This slide doesn't contain enough information. That's why we're moving to the next slide. Ion exchange chromatography is a technique that takes advantage of the differences in the net electric charge of proteins at a specific pH. The key idea here is that proteins have different charge properties depending on the pH of the environment, which influences how they interact with the chromatography column. The column itself is made of a synthetic polymer, often called resin, which contains charged groups attached to it. These charged groups are what enable the separation. If the resin has bound anionic groups, we call it a cation exchanger because it will attract positively charged proteins. Conversely, if the resin has bound cationic groups, it's an anion exchanger, which interacts with negatively charged proteins. Now, the affinity of each protein for these charged groups depends on a couple of factors. First, the pH of the solution, since it determines whether the protein is positively, negatively, or neutrally charged. Second, the concentration of free salt ions in the solution, which compete with the proteins for binding sites. So, by adjusting the pH and salt concentration, we can selectively bind and then elute proteins based on their charge properties. This technique is powerful because it allows us to separate proteins that might be very similar in size, but differ in their charge characteristics, connecting nicely to our overall theme of protein purification and analysis. Separation can be optimized by gradually changing the pH and/or salt concentration of the mobile phase, which creates what's called a pH or salt gradient. This gradual change helps us better separate proteins based on their unique properties. In cation exchange chromatography, proteins that carry a net positive charge tend to migrate through the matrix more slowly than those with a net negative charge. That's because the positively charged proteins interact more strongly with the stationary phase, which slows them down. As we continue, remember that ion exchange chromatography, especially in this context, relies heavily on these charge interactions. By adjusting the pH or salt levels, we can fine-tune how proteins interact with the stationary phase, making our separation more efficient and precise. This technique is a powerful tool for isolating specific proteins in complex mixtures, connecting directly to our overall goal of achieving high-quality purification. This slide doesn't contain enough information. That's why we're moving to the next slide. Size exclusion chromatography, also known as gel filtration, is a technique we use to separate proteins based on their size. In this method, you'll find that larger proteins actually come out of the column before smaller ones, which might seem a bit counterintuitive at first. The key to understanding this lies in the solid phase of the column, which is made up of cross-linked polymer beads. These beads have specially engineered pores or cavities of a specific size. Now, larger proteins are too big to enter these pores, so they can't go inside the beads. Instead, they travel around them, taking a shorter and faster path through the column. This means that the separation is based on size, with the larger proteins eluting earlier because they don't get trapped in the pores, unlike the smaller proteins that enter the cavities and take longer to come out. This technique is quite powerful for analyzing protein mixtures and understanding the size distribution of proteins in a sample, fitting into the broader theme of protein characterization and purification. Small proteins enter the columns and tend to move through the cavities, but they are slowed down because their path is more complex and labyrinthine. This means that their journey isn't straightforward, and they take longer to pass through the column. This characteristic is actually useful because size exclusion chromatography can be used to estimate the size of a protein being purified. By observing how long it takes for a protein to elute, we can get an idea of its size relative to other molecules. This technique is especially helpful when we're trying to characterize or compare proteins during purification. So, in essence, size exclusion chromatography not only separates proteins based on size, but also gives us valuable information about their approximate size. This ties into our broader theme of understanding protein properties and behavior during purification processes. This slide doesn't contain enough information. That's why we're moving to the next slide. Affinity chromatography is based on binding affinity, which means it leverages the specific interactions between a molecule and a target protein. The beads in the column are covalently attached to a chemical group called a ligand, which is a molecule that can bind to a particular protein or macromolecule. When we pass a mixture of proteins through the column, only the proteins with an affinity for that ligand will bind to the beads, causing their movement through the column to slow down. For example, if a protein's biological function involves binding to ATP, we can attach a molecule that resembles ATP to the beads. This creates an affinity matrix that helps us selectively purify that specific protein. Proteins that do not interact with ATP will flow through the column more quickly. To retrieve the bound proteins, we then elute them using a solution with a high salt concentration or a free ligand, such as ATP or an analog, which competes with the bead-bound ligand. This method is powerful because it allows us to target specific proteins based on their binding properties, making it a valuable tool in protein purification. Salt plays a role in weakening the binding between the protein and the immobilized ligand, mainly by interfering with ionic interactions. When we add free ligand into the system, it competes with the ligand attached to the beads, which causes the protein to be released from the matrix. Often, the protein that comes out of the column is still bound to the ligand used for elution, which is something we need to keep in mind. In many purification protocols, we use genetic engineering to add extra amino acids or peptides, what we call tags, to the target protein. These tags are really useful because affinity chromatography can specifically bind to them, allowing us to achieve a significant increase in purity with just a single step. Another advantage is that, in many cases, we can remove the tag after purification, which helps restore the native function of the protein completely. So, to sum up, affinity chromatography is a powerful technique, especially when combined with tagging strategies, as it simplifies purification and can preserve the protein's natural activity. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide is about a specific type of chromatography called high-performance liquid chromatography, or HPLC for short. Essentially, in this method, we use a liquid, known as the mobile phase, to push the sample through a column at high pressure. The column itself is packed with a stationary phase, which helps separate the different components of the sample. What makes HPLC stand out is the use of high-pressure pumps. These pumps accelerate the movement of protein molecules down the column, making the process much faster and more efficient than traditional chromatography methods. Additionally, HPLC utilizes higher-quality chromatographic materials that can withstand the crushing force generated by this pressurized flow. So, in summary, HPLC is a powerful technique that combines high pressure, advanced materials, and precise control to achieve rapid and effective separation of complex mixtures. This method is especially valuable in research and industry, where speed and accuracy are crucial. It really highlights how technological advancements have improved our ability to analyze and understand biological molecules. By reducing the transit time on the column, HPLC can limit diffusional spreading of protein bands and thus can greatly improve resolution. This means that when the molecules move through the column faster, they don't have as much time to spread out, which leads to sharper, more distinct separation. This technique yields perfect results in the separation and identification of a wide range of molecules, including amino acids, carbohydrates, lipids, nucleic acids, proteins, steroids, and many other biologically active molecules. In HPLC, the mobile phase passes through the columns under a pressure range of about 10 to 400 atmospheres. This high pressure helps push the mobile phase and the sample through the column efficiently. Additionally, the flow rate of the mobile phase is quite high, typically between 0.1 and 5 centimeters per second. This high flow rate contributes to faster analysis times without sacrificing resolution, making HPLC a powerful and versatile technique for separating complex mixtures. Overall, the ability to precisely control flow and pressure conditions allows HPLC to achieve highly accurate and rapid separations, which is essential in many biochemical and clinical applications. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a moment to review the key components that make up an HPLC system. Each part plays a vital role in ensuring accurate and efficient separation of compounds. First, we have the solvent reservoir, which holds the mobile phase, essentially the carrier liquid that moves through the system. Next, the pump is responsible for pushing this mobile phase through the entire setup at a consistent flow rate, which is crucial for reproducibility. The injector then introduces the sample into the mobile phase stream, allowing it to be carried into the column. Speaking of the column, this is the heart of HPLC where the actual separation occurs, based on interactions between the sample components and the stationary phase. The detector follows, which identifies and quantifies the separated compounds as they elute from the column. Finally, the data system records and analyzes the signals received from the detector, providing us with detailed information about our sample. When you understand how each of these components connects and works together, it gives you a clearer picture of how HPLC achieves precise and reliable analytical results.